Hello, and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today we'll be talking to Dr. Gabriel Nuto Nóbrega about his paper, Diffuse reflectance spectroscopy, vis nurse were, as a promising tool for blue carbon quantification in mangrove soils, a case of study in tropical semi-arid climatic conditions, published in the Soil Science Society of America journal. I'm your host, Abby Morrison. Let's talk about science. Hi, Gabriel. Welcome to the show. How are you doing today?
Hi, I'm very fine, and I'm very excited to just talk about mangroves.
Oh, me too. Welcome to the show. So why don't we start, and you can just tell me a little bit about yourself, kind of your background, and how you got to your science.
Okay, my name is Gabriel Nuto Nóbrega. I'm from Brazil. I'm from a city in the northeast of Brazil. It's called Fortaleza. I have my undergrad course and my master's. I got there in agronomy, so I'm an agronomist. And last year I obtained my PhD level at the University of São Paulo in Piracicaba, Brazil. Since my undergrad course, I started working with mangrove soils with Professor Thiago Osório Ferreira as a coordinator and Professor José Luis Otero from Santiago de Compostela as a co-coordinator. Despite being an agronomist, which is a profession that is most focused on agriculture, I think the studies that I took from my university helped me a lot to study coastal wetlands. So this is my background.
Sure. And we're talking about blue carbon and diffuse reflectance spectroscopy today. Can you tell me a little bit more about both of those?
Since I started reading and studying mangroves, I got fascinated by this ecosystem. The beginning of 2000s, like I think 2007, mangroves got highlighted by its importance as a globally significant blue carbon sinks, which means that these ecosystems are very capable to sequester carbon, to take carbon from the atmosphere and store on the soil. But there's a problem for the quantification of the carbon stored in mangrove soils. Since mangroves are often flooded soils from the tidal, the soils have particular characteristics that the commonly used method is not a proper manner. The commonly used methods for quantification of carbon in mangrove soils, they normally overestimate the carbon content, which is not adequate. So we started thinking which methods could be used to quantify carbon in mangrove soils. Then we started using diffusive reflectance spectroscopy, which is a technique that we put a light in our sample and we use a spectrometer, or we use a device that captures the light that reflects from the sample, and using the curves that we obtain from this light, we can quantify carbon. So it's a much more easier, it's a cleaner way to quantify carbon, and it's much more cheap than the other ways. It doesn't have the problems that the chemical methods for carbon quantification have. So it's been a very promising tool. I just must say that diffusive reflectance spectroscopy is being used for soil sciencing, have been used during the last, I think, 15 years. So we try to adopt this technique for coastal wetland soils as mangroves.
Sure. So can you tell me some of the problems with kind of the more traditional methods? Like, why don't they work on mangrove soils as opposed to other soils?
Sure. I'm going to try to speak it in a more comprehensible way, I think. In terrestrial soils, like this, in soils that are not flooded, The microorganisms, as we do, use oxygen for their respiration to obtain energy. When we have fluid conditions, this kind of respiration doesn't occur. So the microorganisms must use another compounds to breathe and to obtain energy. So in fluid soils, they started using some nitrate, some iron, some manganese, and some sulfate, which is very abundant in mangrove soils. The problem is with these metabolic pathways, with these respiration pathways, some reduced compounds that we say are produced. So when we try to quantify carbon, we measure something else than carbon. So that's the major problem for the quantification itself. But when you start thinking that for this carbon quantification, use strong acids, you start using strong oxidizers, we generate a lot of residue, which is very toxic. We use chromium, which is a toxic metal. So these are the other advantage of chemically carbon quantification.
Sure. So you had a good analogy to describe diffuse reflectance spectroscopy, which I am going to call DRS from now on because it's very hard for me to say for some reason. So can you tell me kind of an easy way to understand what that technology does?
Sure. The diffuse reflectance spectroscopy, or DRS, it's a method, it's a technique that works basically as we see the world. In our world, we have a big lamp, which is the sun, that emits lights. The lights travel through the universe and interact with the object. The objects are then reflected to our eye, and that's how we see the things. For example, a flower. For the diffuse reflectance spectroscopy, it's the same thing, but instead of using this natural light, we use a specific light in a dark room that we light our samples, and then we collect the light that is reflected by the sample. And using this light, we can quantify some properties of the objects, for example, carbon.
Excellent. Okay, so we have this environment, mangrove forest. Can you tell me a little bit what that looks like?
Sure. Mangroves, it's a typically tropical ecosystem that extend from the tropical regions of all over the globe. And it's basically made of trees that are adapted to the tidal fluctuation and the salinity fluctuation that occurs. For mangrove soils are in that coastal zone that the sea water rises and flows those fields, and then when the tidal return to its lower levels, it's increase the salinity on those areas, and just mangroves are basically mangroves are able to grow on those sites. They are commonly sticky soils with a lot of carbon, and some animals that are adapted to live with those trees. For example, crabs, some birds. Due to the fact that the mangroves are located in a transitional zone from the dry upland areas into the ocean, it provides a lot of ecological function. For example, mangroves are very important to act as a nursery for birds, for fish, for mammals like dugongs and some dolphins as well. So in summary, I could say that mangroves are one of the most important forests in the world, which are basically made by trees and that are adapted to live with a wide salinity range in flooded soils and to those reducing conditions that are characteristics of these ecosystems.
Great. So you had these forests and you wanted to see if DRS would be able to correctly gauge how much carbon is in them. So let's move on to kind of how you did that. Can you tell me how you went and gathered your samples, what you were looking for in a sample, anything like that?
Okay. During my master's, we started working with carbon quantification in mangroves. The main focus of my work was to understand how carbon dynamics occur in semi-arid mangroves. So, for those samples that we took for our study, we sampled in 3 different mangroves along the Ceará coast, which is a state from Brazil. Then we sampled in each mangroves. We established some sampling points. We tried to take points at the same physiographic conditions. Mostly of those sites were based on the same tree species. And then we collected samples using a sampler, which is a stainless steel tube pipe that we insert another pipe inside to take the samples. Like, so we take this stainless steel pipe, we insert it into the soil, then we take some PVC tubes, put it inside, close it, and take the samples. I can say one thing, that it's not very easy to walk in those mangroves. We got some friends that got a little bit stuck because mangrove soils, the mangrove soils are a little bit sticky, and it's not as hard as the common soils. So sometimes you can get a little bit stuck, but it's part of the work. And by the end of the day, it's a lot of fun that we have there. It's like we're looking always for most protected mangroves areas, so we have a lot of beautiful sites to sample.
Sure.
So it's a good experience to see some good preserved areas and something that are not common for everybody. Not everybody have the opportunity to see such a beautiful forest as mangroves.
Yeah.
So it's very pleasure. And also sometimes when we are sample, we can see some manatees. I've seen a lot of turtles. We see a lot of crabs. It's always fun to see the nature, how it works, and how beautiful it is.
Sure. Yeah, that sounds beautiful.
It's really beautiful.
So once you got your samples, how did you transport those? I mean, is it something like sand art or like a soil profile where if you shake it up, you're going to ruin it, or does it not matter?
As we collect in PVC tubes, we have some caps that we close those PVCs. We put those samples in refrigerated box because we are also interested to comprehend how the reduced compound drives carbon dynamics. We put the samples in an insulated box, transport in a vertical position, because first we have to be sure what is the upper side and the deeper samples. So that's how we have to do. The problem is sometimes we have to put the samples in an insulated box inside the mangroves. So we have to carry all our stuff inside the mangrove, make the sampling, put it in an insulated box, and transport sometimes by boat, sometimes by walking.
Wow.
And it's probably the hardest part of the job.
Sure.
Then when we got at the lab, we take our samples from the PVC tubes using a device like a syringe. We insert a smaller PVC inside the tube to take the samples from the PVC. We take some subsamples that we freeze for other analysis. And for carbon quantification, we dry the samples, we remove the inorganic carbon that might be from salt precipitation or from shells. Then we quantify carbon by elemental analyzer. For reflectance spectroscopy, we just need to dry the samples, grind it a little bit to make it more homogeneous, and then we analyze.
So each of these samples, are you checking by like how much carbon is in, I don't know, the top foot or however much distance, and then how much is in the lower, or is it just it doesn't matter?
For the DRS study, for us, it doesn't really matter. We just measured for those soil layers because we also would like to characterize the areas.
Sure.
Because we took 3 very different sites to see the wide range of the carbon contents from the semi-arid mangrove soils. So that's why we took samples from different depths, but it's just because to take a better representation for our mangroves.
Okay. So then what did you find? Did it work as you hoped? Were there any surprises?
Yeah, in fact, it worked really well. We got some statistical models that show that it's a very promising tool. Also, there's the opportunity to use reflectance spectroscopy to check the other characteristics of mangrove soils. And in probably a half day of working and taking the light spectrum, we could do like 100 of samples, which we are not able to do using chemically methods. So it's a much faster way to quantify carbon.
Yeah.
It's a much more easier way.
Well, that's great. Congratulations.
Thank you.
So tell me kind of what some of the practical implications of this research. Where else can other scientists use this, and why is it so crucial that research like this is being done?
Okay. So, as I said, since mangroves have been highlighted by its ability to sequester carbon, to store carbon on its soils, some policies have been made in order to give money for those who protect mangroves and who restore mangroves. So in restoration practice for mangroves, one of the crucial things to get, for example, carbon credits, which is a money that you can get for carbon sequestration, you must quantify the carbon stocks in an adequate way. So, as we are getting a faster and a very adequate method for carbon quantification, the management of these restored areas and the implementation of restoration programs for mangrove soils is going to be much easier, and it's going to be cheaper because you're going to have to use less money to monitor your carbon stocks. We also without producing toxic wastes.
Sure. And what kind of research is your team looking to do after this? What's on the horizon for you?
So our research group have been studying coastal wetlands for at least 10 years now. We've been studying mangroves, which most of our work, but we also have been studying hypersaline tidal flats, seagrass meadows, salt marshes, coastal lagoons, and all those coastal wetlands in order to better understand the cycle of the elements in these ecosystems. Most recently, we started working with mangroves recovery process, with mangroves reforestation. People are getting known with the importance of mangroves and then started to replant those forests. And one of my research project that is now going on is to identify and to quantify the dynamic of this process with within replanted mangroves. So we want to see for how long we must take care of replanted mangroves to see that they are now stable and well-developed forests. So we can, with these two information, a better way to quantify carbon in the time that is necessary for the reestablishment of mangroves, we think that it's going to be a great move forward for mangrove science and for mangrove protection, since we could measure better the efforts for mangrove reforestation.
Sure. All right. I have 3 questions left for you. First question. If people want to learn more about blue carbon and carbon sequestration, where can they go?
Sure. There's a lot of nice and very beautiful websites on the internet that you can find a lot of valuable information about blue carbon sinks, and others. There's a very beautiful website which is the Blue Carbon Initiative.org, and there's the Blue Carbon Portal, which is bluecarbonportal.org. Also, on the internet, there's a lot of web books or e-books that you can take for all the people. There are a lot of books for children. There are a lot of books for people who are not very familiar with biogeochemistry, but where they can have a lot of very nice information. If you want some information about our research group, you can go to Twitter and look for Gabriel Nóbrega, which is me. You can go to look to see Thiago_O Ferreira. Also, there is Queiroz HM and Danilo_Iche, ICHE, which are some of the researchers from our study group. Also, you could see the website of our department, which is Soil Science Department from the University of São Paulo.
Perfect. We'll include links to all those on our show notes as well.
Okay.
And then second question is, if people are interested in kind of some of these topics, carbon sequestration, mangrove restoration, what steps can they take to get involved in that process or to kind of help with some of those issues?
For those people who are interested to help blue carbon initiatives and coastal wetlands protection, I could say that there are a lot of non-governmental organizations that protect coastal habitats. Also, we have some museums in Brazil that are basically directed to those ecosystems, as we have some mangrove museums here. But also, I think it's an important thing to say is that we must also think about our acts with wastes and consumption, since as we are looking, a lot of the garbage that we throw away, they most likely to go to the sea and then return to mangroves. So it's into the other coastal wetland as well. So I think that's something that we must do. Also, maybe join one of those non-governmental organizations.
Sure. Yeah. Did you see a lot of waste items as you were going out to collect your samples, or?
Yeah, unfortunately, it's been very common when we're going to take some samples from mangroves, also from seagrass. We see a lot of plastics, and also it's important to aware people from this huge problem, and it's globally spread plastics.
Yeah, they're a big problem. So, final question for you.
Okay.
So this is kind of the fun question. Well, I mean, they're all fun questions. But what is one fun fact about you that people would not know if all they had was your research?
About me?
Yeah.
Oh, that's a tricky one. So, the most things that I like to do are always linked to the oceans and to the coastal zones. I think that's why I love mangroves and other coastal wetlands. Because since a kid, I started to go traveling to the beach. I tried and I learned how to surf, to spend more time on the ocean. I learned how to windsurf. I love to travel, to do some snorkeling and some scuba diving. So I would say that I'm a person very close to the ocean, despite being an agronomist and learn how to cultivate corn. I would say that.
All right. Well, thank you so much for your time, Gabriel. I really appreciate you taking some time out of your day to be on the show. It's been so fun to talk to you.
It's a pleasure to be here talking with you and to those who are listening. And I hope I could help to the protection of those sites and to show to the audience that we can do a lot of good science outside the chemical lab and to enjoy the nature as we are doing some research. It's one of the best things we can do.
Yeah, for sure. Well, thank you.
You're welcome.
Thank you for listening to Field Lab Earth. If you'd like to find out more about today's topic, our featured article for this episode will be freely available for the next 2 weeks. You'll find a link to today's paper and other resources for this episode in our show notes or on our website. If you have any questions, comments, or recommendations for show topics, please contact us at podcast@sciencesocieties.org or on Twitter @FieldLabEarth. If you'd like to hear more content like this, please subscribe, and don't forget to rate and review us on iTunes or anywhere else you find your podcast if you like our show. This podcast is a joint production of the Tri-Societies, the American Society of Agronomy, Crop Science Society of America, and Soil Science Society of America. Special thanks to Lobo Loco for the use of their song Spook Castle on the intro and outro of our show. Opinions and conclusions expressed by authors are their own and are not considered as those of the American Society of Agronomy, Crop Science Society of America, Soil Science Society of America, its staff, its members, or its advertisers.